Hi friends, so now I am back with my next video, and in this video we are going to talk about translation in prokaryotes. So in my previous video I have talked about replication, transcription in prokaryotes and eukaryotes, and today we are going to talk about translation, which is the synthesis of protein in prokaryotes. Please make sure to like, share and subscribe my channel. So what is translation? It is the process of protein synthesis in which the mRNA get translated to a protein, which is a sequence of amino acids. So here you can see that from DNA to RNA, this process is called transcription, and from RNA to protein, it's called translation, in which the protein polypeptides are formed. And it always, like the protein synthesis, it proceeds from the N-terminus to the C-terminus, and ribosomes are read in the 5 to 3 direction. So suppose you have an RNA, so it is always read from 5 to 3 direction, and it is synthesized from N-terminus to C-terminus. And translation, it takes place in the ribosomes, which are present in the cytoplasm of the cells. And in the prokaryotes, as long as the proteins, like the transcription is happening, like the from DNA to RNA, the transcription is happening and the strand is getting released, and the translation starts at the same time. But for eukaryotes, the process is a bit different, because the RNA that is released, it needs to go into further processing like splicing, which I have talked in my previous video, and different modification, and then only it can proceed to the translation in the cytoplasm, because in eukaryotes the transcription occurs in the nucleus and then it get transported to the cytoplasm where translation can take place. But in prokaryotes, this process occurs in the cytoplasm. So, what are the different components which are required for translation? So, as I have talked, the most important component is the ribosome, where the translation takes place. So, ribosomes, they are basically distributed throughout the cells, and they are approximately like half protein, half RNA. So, here you can see the ribosome. It consists of two halves: a larger subunit which is 50S and the smaller subunit which is 30S. And as you can see, they contain 16S RNA and then protein, and same here, different rRNAs, ribosomal RNA and protein. So they are composed of half protein, half RNA, and when they need to do the translation, the both subunit assembles together like a sandwich, and in between is the mRNA. So here you can see the different subunit, the small subunit and the larger subunit. So the smaller subunit, it binds to the mRNA, and once it is bind, and to elongate the chain, like to initiate the translation, the larger subunit, it binds for the it binds to the tRNA, which brings the amino acids which are and which have the anticodon to the mRNA, and then also catalyze the formation of the peptide bond. So the important thing is that the smaller subunit is responsible for binding to the mRNA, and then further process like the different tRNAs, different sites for binding are present in the larger subunit. So what is tRNA? We have talked about different tRNA. What does they do? So what is tRNA? So the message, like the information which is present in the mRNA, it is encoded, like decoded by the tRNA, and it brings the specific tRNA molecule which has the anticodon for the mRNA. And so at least like one specific tRNA is present for a per amino acid, and because different codons can code for one amino acids, so the tRNA can recognize these codons but only for one specific amino acid. And the anticodon arm of the tRNA, as I have talked about, they contains a triplet nucleotide sequence which is complementary to and base pairs with the mRNA. So suppose you have AUG at the mRNA, so in the tRNA it has to be the opposite, like UAC. Only then this tRNA will come and bind to the mRNA, because then it will form base pairing. So if that base pairing is not matching, that tRNA will not bind to that site and will be removed. So this is very important for the initiation of translation and for continuation, because the base pairing between the tRNA and mRNA has to be there for the translation. And because it carries both amino acid and to recognize the code, and also recognizes the code, it is also known as adapter molecule. So what are different binding sites on a ribosome? So as I was saying, so suppose this is the mRNA, right? And suppose we are talking about UGG or something else. So here you can see this color, this dark red color. This is the tRNA, and here you can see this arm contains exactly the codons, the nucleotides which are complementary to this. So if this is not complementary, this tRNA cannot be present here, right? So this anticodon arm has to be complementary to this mRNA sequence, the three nucleotides. So there are basically three sites in the ribosome which can bind to tRNA: E site, P site, and A site. So what is A site? A site is also called the amino acyl site, which accepts the tRNA. So suppose we are talking about, like, we are at this level like UGG. Now we move to UUC. So this site accepts the incoming tRNA for the next amino acids to be added. So now it contains the tRNA for this UUC, which is AAG phenylalanine. So this accepts the tRNA for the next amino acid. Then is the P site or the peptide site, which binds to the tRNA and the growing polypeptide chain. So suppose we are starting from here, P site is the one which binds to the polypeptide chain and the tRNA which contains the polypeptide chain. And then E is the exit site where this amino acid get transferred, and what left is the tRNA without the amino acid, which is called the uncharged tRNA. So here you can see there is no amino acid, like you can see here is the phenylalanine, but here there is nothing. So this is called uncharged tRNA. And then exit site is the site where the uncharged tRNA binds and then get released from here. So these are the three important sites which you need to know. And then there are other components which are required, like obviously the messenger RNA, which act as a template for the synthesis of polypeptide chain, amino acids, so that they can be charged with tRNA and can be used for the protein synthesis, ATP, GTP, which are required for the source of energy, and their hydrolysis is important for binding of some factors. And then there are different factors which are required for initiation, elongation, and termination, which I will talk about in my video. So first step is the activation of amino acids or the charging of tRNA. So what happens is the tRNA is not bind to amino acid in the normal state. But when the attachment of amino acids to tRNA occurs, this is occurred by the enzyme amino acyl tRNA synthetase. And then the tRNA which is formed with amino acid is called the amino acyl tRNA. And then the amino acids are attached by the high energy bond between the amino acids and the tRNA. So here you can see we have the enzyme amino acyl tRNA synthetase, and then we have amino acids and the ATP. So these amino acids, they bind to this enzyme, and then with the help of the phosphate bond of adenine, pyrophosphate is released, but this adenyl phosphate remains here with the amino acids. And then the tRNA, which is anticodon to this amino acid, it binds to this site, like, and binds, and then this AMP get released, and also what it get released is the charged tRNA. It's called charge because this tRNA is bind to the amino acid, and then the enzyme gets released. So this is called charging of tRNA. So how does the charged tRNA looks? You can see it has the amino group, amino acids, which can be anything which needs to be complement anticodon to this one, right? And then like which can code for this amino acid, and then it is attached to the ribose adenine. So as you can see here, the with the adenine group, right, it is attached to the adenine group of the tRNA chain, right, ribose of the nucleotide of tRNA chain. It is attached by the carboxyl group, and always there are this CC cytosine nucleotide which are present, and then this loop structure which is contains the anticodon for mRNA. So this is how the charged tRNA looks like. So now we talk about the initiation. So it starts with the binding of the like recognizing the site where does the initiation of the translation takes place. So it starts with the with always same amino acid methionine or formyl methionine, which is the formylated version of methionine. So in eubacteria such as E. coli, AUG or GUG or sometime UUG, they serve as the initiation codon, and these because they code for methionine, and they are recognized by N-formyl methionine tRNA. So here you can see the formylated amino acid methionine which is formylated, and then the important difference, because this is the initiator tRNA, because not anywhere you will find the formylated amino acid, because this is the first amino acid which is recruited. So the important difference between the initiator tRNA and other tRNA is this CA, which does not form the bond, and this functions in the formylation, and then this is the loop structure of tRNA, and there are three GC pairs which allow entry to the P site. So as I have talked about, usually the tRNA that comes, it comes at the A site which accepts the tRNA, but during the initiation the tRNA directly binds to the to the peptidal site, which this GC helps in binding of this formyl tRNA to the P site. So how does the ribosome recognize where to bind in the tRNA in the mRNA? So it is done by the Shine-Dalgarno sequence, which is a sequence present in the prokaryotic mRNA, which is located generally 8 bases upstream of the AUG, and this sequence helps to recognize the site and makes the ribosome binds to the mRNA. And then this 30s subunit, it contains 16s rRNA, which contains a sequence which is complementary to this Shine-Dalgarno sequence. So here you can see this is the mRNA, and 8 base pair upstream of the mRNA is the Shine-Dalgarno sequence, and then 16s subunit, 16s rRNA of the 30s subunit contains a sequence which is complementary to this Shine-Dalgarno sequence, and this is the place where the ribosome subunit binds and initiates the translation. So initiation, basically there are different factors involved as I have talked about. So here is the small subunit, and you can see there are different sites E, P and A. So first is IF1 and IF3. They bind to the 30s subunit and prevents the larger subunit to bind. And then this IF2 GTP binds, which will help that. So IF2 GTP binds to the peptidal tRNA, and it will help the initiated tRNA to bind later. So here you can see we have the IF1 and IF3 binds to the 30s subunit, and then it binds to the mRNA, and then IF2 is bind to the P site. When the tRNA comes, this binds to this site, and the IF1 and IF3 are released, and by the GTP hydrolysis this IF2 factor is released. So because IF1 and IF3 get released, the larger subunit can bind, and now the initiator tRNA is at the P site. So we have our first tRNA which is bind to the P site. Now the step is to binds to another tRNA. So as I have talked about, in which it's a cyclic process in which one amino acid is added at one time, and it begins with the binding of the second amino acid tRNA at the A site. As I have told you that they accept the new tRNA at the A site, which And then there are different factors which are involved. EFTU, which directs the tRNA to bind to the correct position, like at the A site, and then because then it get released and then it is regenerated by the GTP by EFTS, and then EFG mediate translocation, which I will talk about what is mean by translocation. So here you see the first tRNA that we got with the P site. So next step is to get this UUU, which is coded by the phenyl NNA. So now what we get is this EFTU factor, which is bind to GTP and the tRNA. They come and binds to the A site, and EFTU is hydrolyzed and AFTU GTP is released. So now we have two tRNAs, one at P site and one at A site. So what happens is the translocation. What does that mean? So this formyl methionine or methionine, which is in orange, you can see it moves to the A site without the tRNA. So only the amino acids from P site moves to the A site, and then what leaves is the tRNA without the amino acid, which is the uncharged tRNA. So now, because if the next amino acid, next tRNA comes, we need this site empty. And so what happens is the translocation. So during translocation, this uncharged tRNA, it moves to the E site, the exit site, and then this tRNA with two amino acids moves to the P site, and this happens by different factor, which is EFG GTP, and it uses hydrolysis as an energy source to move this tRNA. So now when the next amino acid, next tRNA comes, we have the A site empty, and the same process is continues until we reach termination. So this is how the elongation happens. So now is how we know where is the termination. So it depends when a stop codon arrive at the A site. So stop codons are mainly UAG, UAA and UGA. So these three are mainly the stop codons, which when they arrive at the A site, it is recognized and bind by the release factors. So what does release factor do? They bind to the termination codon at the A site and stimulate the hydrolysis and between, like, breaking the bond of the tRNA and polypeptide chain, and then they both are released, and then the release of the mRNA molecule also occur, and the two subunit get separated. So here you can see now this A site release recognize the stop codon, and then release factor binds here. So once the release factor binds, they release the free polypeptide and the tRNA molecule from here, and then the mRNA molecule get released, and the two subunit get separated. So all this occur by the hydrolysis of GTP. So I have talked about initiation, different sequence, Shine-Delgarno sequence, how does the ribosome know where to start the translation, how the elongation occurs by translocation, different sites E, P and A, and then the termination. I hope it's clear. If not, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.